Associate Professor Dr. Sarina Sulong, who will discuss the current application on microarray technology. Okay. But permit me to introduce Associate Professor Dr. Sarina Sulong for a few words. So fairly speaking, I would say that Associate Professor Dr. Sarina Sulong, she's a really well-known person in our school when it comes to choosing who to speak with about genetic research. All right. So from University Kebangsaan Malaysia to University of Science Malaysia and then to Newcastle University of United Kingdom. So those are actually describing her educational background, with Dr. Sarina having a specialty in human cancer genetics. Experience-wise, I would say Associate Professor Dr. Sarina Sulong has almost 20 years of hands-on experience in numerous genomic research techniques that include SNP array, single nucleotide polymorphism array, gene expression array, qPCR, quantitative real-time PCR, as well as FISH, fluorescence in situ hybridization. And she's currently a coordinator, or we call it penyelaras, at Human Genome Center, School of Medical Sciences. And right now, Associate Professor Dr. Sarina Sulong has supervised and graduated around 20 postgrad students that include M-Science and also PhD students. And her work has been published in a number of highly regarded research publications. So with her today, we want to learn, we hope that we can learn more about what is the advancements of a gene expression array and how the technique can be considered as a critical method for studying cancer genetics. So I would say this is quite very interesting topic indeed. And I think our speaker, Dr. Sarina, are you ready? So yes. Without further ado, let's welcome Associate Professor Dr. Sarina to share her talk.
Okay, let me share my slides first, okay? Okay, can you see my slides?
Yep, Dr. Sarina, yes.
Okay. Okay, Bismillahirrahmanirrahim. Assalamualaikum and good morning. Thanks to our moderator, Dr. Norasikin, for your kind introduction. And thanks again to Dr. Shazana Hilda and also Department of Pathology for inviting me to be one of the speakers in this webinar series. My presentation will be focusing on microarray technology in biomedical research, particularly in genetics or genomic research. And I will introduce the general information about microarray, and then focusing on DNA microarray, its application, and a few things related with microarray data analysis. Okay, so first of all, let me introduce a little bit about biomedical research, as actually my undergraduates or my first degree actually in biomedical sciences, okay, from UKM. So biomedical research is an approach used towards solving medical problems. So it is actually related with medical and also health. So it is also deals with beliefs or theories which can be proven or disproved through observations and experimentations. So based on these States United for Biomedical Research in the USA, so biomedical research also is the area of science devoted to the study of the processes of life, the prevention and treatment of disease, and also the genetic and environmental factors related to disease and health. So there are three different kinds of biomedical research, such as basic or pure research, applied research, and also clinical research. So as most of our clinicians using the clinical trials, so this is also part of the clinical research. And types of models or use in biomedical research may involve human or non-human samples. So it's actually considered as whole living organisms. And also living systems are composed of samples from the original animal or human cells. For example, like we're using the tissue culture, and also non-living mechanical or molecular systems, and also mathematical models. For example, like computer simulations also is actually part of the biomedical research. So in basic biomedical research, usually we do using or doing the web experiments in the laboratory. And the focus of study may involve human anatomy, biology, and also physiology in relation to disease and organ tissues, and also focusing on the cellular or molecular or genetic levels. And the research goal actually is to generate new knowledge that explains biological phenomena. So since the word of genetics is commonly stated in biomedical research scope, and also genetics is also part of my research field, so I think it is relevant to introduce these common terms like genome, genomics, and genetics. So genomes, as we all know, that is an organism's complete set of DNA. So the complete set of genetic information in an organism, such as plants, virus, human, bacteria, or animals. And what is genomics? Genomics is the study of whole genomes of organisms and incorporates elements from genetics, and also the study of all of a person's genes, including interactions of those genes with each other and also with the person environment. And genetics actually is the study of heredity, the process of a parent passing certain genes to their children. So most of the contents in my presentations actually will be related with these terms. So before we go into details about microarray, let me ask you actually, why are genetics and genomics important to our health? So genetics and genomics both play roles in health and disease, and both may provide more understanding about diseases, including complex diseases, which can lead to earlier diagnosis, interventions, and also targeted treatments. Genomics includes the study scientific of complex diseases such as heart disease, asthma, diabetes, and cancer, because these diseases actually are typically caused by more a combination of genetics and environmental factors than by individual genes. And genomics is offering new possibilities for therapies and treatments for some complex diseases, as well as new diagnostic methods. So in the literature, it has been reported that genomic technologies or approaches, including this microarray, actually are important for understanding the genetics of complex disease, also may have potential impact in the clinic, especially for diagnosis and patient management. And genomic technologies may provide important data for precision medicine and may discover various novel findings for further understanding of a genetic disease. So the Human Genome Project, completed in April 2003, gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. It allows more research to be done to fully understand how the genome functions and to discover the genetic basis for health and disease. And these are the common genomic approaches that are being done, particularly in research, including genome sequencing, genome-wide association studies, epigenomics, and also gene expression profiling. So it has been reported that genome-wide association studies, or GWAS, became feasible and made a major contribution to understand the genetics of complex traits and also diseases better. So of course, we can use microarray to do the GWAS studies. So what is a microarray? Microarray is a laboratory tool used to analyze large numbers of genes or proteins at one time. So in microarray, biologic molecules such as DNA, cDNA from the mRNA, or protein are placed in a pattern onto a surface such as a glass slide. And then we need other substances which are added to these slides to detect specific patterns of molecules. And then most microarray protocols use reverse transcriptase to convert mRNA into cDNA because DNA is more stable than RNA. And the number of different genes or nucleic acid sequences that can be simultaneously assayed on one DNA chip can range from 10 to 1 million or more. And this technology works under the principle of binding, also called hybridizations. So hybridization is one of the main process in the microarray flows. And then the strands of complementary sequences of DNA or cDNA with each other. And microarrays are being used to help diagnose disease, such as cancer, and also to develop treatments for them. Okay, so a little bit about the history. So microarray is created actually based on the studies related with automated arrays and also DNA cloning. So actually the evolution of this microarray actually based on the southern blot. Okay, if you can refer back to the southern blot analysis. So the ability to analyze multiple hybridization signals or hybridization targets was automated in the late 1980s and also early 1990s by using a robotic system to rapidly array clones from microtiter plates onto filters. So in 1995, actually the first study that used the word microarray was published, which is the introduction of miniature microarrays. So in this study, actually, they used the expression of many genes, which can be monitored in parallel. And also the sample array was constructed through higher speed robotic printing of DNA on glass. So you can just imagine just only one glass slide. It's like they're using in the pathology. So it's like one slide that you can just put a lot of the DNA informations on the slides. And in 1997, researchers from Stanford University published the first whole genome microarray study of gene expression by placing the whole yeast genome on a microarray. And starting from that, all the manufacturers, the array manufacturers starting to develop a more and more advanced array as to be used in the research and also diagnostic. Okay, this article was published in 2005. It's about 16 years ago. And it stated that my DNA microarrays or microarray is actually particularly powerful in the context of knowing the entire genome sequence and can provide a global view of changes in gene expression patterns in response to physiologic alterations. So actually, it was firstly started as a gene expression profiling, okay? And then later on moved to the using the DNA samples. And then it is clear that the development of array-based technologies represents a fundamental shift in the way the scientists study living organisms. And this technology is a first glimpse into how researchers will utilize genome data in coming years. So microarray technology is still in a continuous state of evolution and development at that time, because we can see like during that within the 16 years or almost like 20 years, actually, we have a very advancements in this microarray technology. So microarray is currently used to analyze many different systems, and it enables researchers to investigate and analyze the expression of thousands of genes in a single region and address various issues. But not only the expression, but also can see the genetic changes that happen in the at the DNA level. So it is mostly used in cancer research and also in the pharmacological treatment of other diseases, okay, and like different kind of cancer types or whatever. So single nucleotide polymorphism and also gene fusion detections is also commonly used for cancer study. And microarray-based comparative genomic hybridization, or we call as array CGH, is also being used to investigate genetic aberrations in genetic diseases. So array CGH also is one of the is a type of microarray-based or DNA microarray. Okay, so it is very important to know which microarray platform that may be suitable for your research. Researchers should ask actually if microarray technology is really necessary for a specific research application, because as you know that microarray is time consuming and slow to set up, but difficult to validate, expensive, and requires great analytical expertise. So of course you can see like it's automated, but the process also needs trained people to do that, and also of course to analyze is the main part, to analyze the data. Microarray can give an enormous amount of irrelevant data, so it's very important to how that we can manipulate and what is the real data that we can get from the analysis. And the available chip may already have the desired set of gene sequences, so maybe we can have like a targeted that we want to use, or you can customize the chip as well. And the multiplex PCR or a custom chip may be a better method for analysis than employing a genome-wide array. So that's are the considerations that you have to think before you choose whether you want to use microarray or NGS or whatever. sorry. Okay, so a microarray has two broad classifications based on its mode of preparation and also on the types of probes used. So based on the mode of preparation of the array, microarrays can be divided into three types. One is the spotted array on glass. The second one is the in situ synthesized arrays, and the third one is the self-assembled arrays. For the spotted array on glass, actually an array is made on a polylysine coated glass microscope slides, and it allows fluorescent labeling of the samples. So it uses actually the inkjet printing technology. So mostly the example, the most common arrays that use this technology is from Agilent, from the Agilent manufacturer, Agilent company. And in situ synthesized arrays, so these arrays are made by chemical synthesis on a solid substrate. And these arrays are used in expression analysis, genotyping, and also sequencing. And they use the technology of the photolithography. Okay, but I'm not very familiar with the photolithography, but actually the basic behind that is very quite complicated if you want to generate the microarray cell. But the common manufacturer that using this technology is from Heatmaps or currently known as Thermo Fisher. So it's like a gene chip. Okay, the gene chip, but not in the glass like that in the from the Agilent. And in the last category is the self-assembled arrays. So this is like a big chip. They're called it's a big chip because they use the bits deposited on the ends of the array. So Illumina actually is using the big chip technology. Okay, so based on the types of probes used, microarrays okay, so microarrays are 12. There are different types of microarrays, which is based on the types of probe used. Okay, so I am also quite surprised when I found that actually there are many types of microarrays. However, there are a lot of things to explore in this type of microarrays. Okay, so like a DNA microarrays, which is the common one that has been used in our field, in my field. So it's like a cDNA microarrays, or also known as the gene expression arrays, or oligo-DNA microarrays, and also BAC microarrays, and also the SNP microarrays. Okay, the BAC is the bacterial artificial clones. So they use the bacterial artificial clones to prepare the probes that can be put on the arrays. Okay. So there are a lot of things to explore actually. Okay, so in DNA microarrays, actually the common microarray that are being used, okay, in genetics and genomics area. So of course, unfortunately, that I don't have any experience using these kind of microarrays. Okay, so in my presentation, I will focus on this DNA microarray. So for the next this presentation. So of course, there are protein microarrays, peptide, tissue microarrays, cellular microarrays, and quite like so many kinds of microarrays. Okay, so this table shows the general function of common microarrays, such as a DNA microarray, which is to measure gene activity and also genotyping, while like protein expression and interactions profiling can be done using a protein microarray. Okay, so this slide shows the examples of microarray chips which are available for different manufacturers such as Agilent, Illumina, and also Thermo Fisher, or previously known as Heatmaps. So this the technology actually to produce the array may different, as I did mention in my previous slide. So we have the three different kinds of technology, and the sample format is also different. For example, in this gene chip by Heatmaps, okay, you can apply actually one sample for one chip. But in this Illumina chip, you may apply 8 samples for one slide. So you're like a different kind of format, okay? And this is the examples of the tissue microarray. So this table shows some of the differences between microarrays manufactured by Thermo Fisher, Agilent, and also Illumina. Actually, it will be good to understand the purpose of the study, and then you may know which type of microarray that is suitable for your study. So, and it is interesting to know that both microarray from Thermo Fisher and also Illumina actually can detect loss of heterozygosity and also UPD, but Agilent array actually cannot. Okay, and then depends on the markers. Okay, so in Thermo Fisher now currently it's actually almost 6.5 million markers that actually can be put on the chips. Okay, and then but most of the Agilent chip actually we can customize. Okay, we can custom designs based on the markers that we want. Okay, so what is a DNA microarray? So this DNA microarray allows scientists to perform an experiment on thousands of genes at the same time. So actually each spot on a DNA on microarray contains a multiple identical strands of DNA, and the DNA sequence on each spot is unique. Okay, so that means actually each spot can represent like the markers that you want to study. Thousands of spots and are arrayed in orderly rows and columns on a solid surface, usually glass. So it depends on the manufacturer. And the precise location and sequence of each spot is recorded in a computer database. And of course, after that, you have to do it data analysis. Okay, so this slide shows the general, what we call that, just want to show you how the process from the overall microarray technology, the general lifestyle of the microarray analysis, starting from the sample preparation. So this is the example that you want to use the gene expression analysis. Of course, you have to have the RNA samples, and then you have to convert into DNA before applying it into the microarray chip. And these are the three, there are three main instruments in microarray system, which include the high hybridization oven, fluid station. Of course, you have to wash your chips, okay, everything that has been, and also scanner connected to the computer. So once the chip has been scanned, the image will be processed for further data analysis by specific softwares. So this slide shows like the current and emerging technologies in genomics. So microarray is one of these technology, okay, and also including the NGS. And using these arrays, scientists can simultaneously evaluate the expressions of 1,000 genes at the mRNA level, for example, okay, and also at the DNA level. So high throughput microarray analysis provides a powerful tools for the discovery of novel biomarkers, which could possibly be used in the diagnosis and or prognosis of diseases. Okay, so SNP array, single nucleotide polymorphism array, is a type of DNA microarray containing design probes harboring the SNP positions, which is hybridized with fragmented DNA to determine the specific alleles of all SNPs on the array for the hybridized DNA samples. So they use the SNP information that have been put in the DNA array. So this array is considered to be one of high throughput and relatively cost efficient and automated genotyping approaches. Array CGH can also be considered as DNA microarray, but array CGH technique has a different principle compared to SNP array. And nowadays, other than that, also we use the gene expression array or cDNA microarray to do the gene expression profiling analysis. And of course, nowadays, NGS is rapidly being used, particularly in genomic research, and using NGS, an entire human genome actually can be sequenced within a single day. So you know that the different, the purpose that if you want to study the point mutation or you want to study the mutations of the disease, in the disease, so you can use the NGS if you want to see the point mutations. And then it allows for sequencing of DNA or RNA, much more actually cheaply than the previously used Sanger sequencing. Okay, so this slide shows the comparison of marker distribution in different arrays and platform in which a cytoscan actually represents as a DNA microarray. Cytoscan actually manufactured by Thermo Fisher, and WES, the whole exome sequencing, and clinical exome actually represent the NGS platform. So in this figure, Cytoscan HD has the highest markers, which is almost 2.6 million markers. as compared to Cytoscan Optima, which is only has 315,000 markers. So if there is a less markers, that means you will see gaps between the regions along the chromosome view. So this is the example like for the deletion of chromosome 4p. So you can see clearly using the Cytoscan HD, there is a deletion on this chromosome 4p. Okay. So this figure shows the schematic overview of the applications of microarray technologies in cancer medicine. And given its excellent performance in detecting predictive genetic biomarkers in clinical oncology, application of microarray technologies to cancer medicine could be a logical approach toward establishing a better management of cancer patients. Okay, and furthermore, the ongoing technical advancements and the growing databases of disease-specific profiles will enhance the current cancer diagnosis, prognosis, and treatment paradigms, as well as assist physicians or clinicians in delivering personalized treatment plans in order to minimize treatment-related toxicity and to improve prognosis. So as we all know that we have the targeted treatment, so we need the information from the microarray, and then they can help clinicians to see whether which treatments that can be targeted for that specific alterations in our patients. So this is one of the applications, like of course identify genes and pathways involved in oncogenesis. So mostly we are using the gene expression profile to see the pathway, and also to identify molecular targets for therapeutics. And we can see the classifications, diagnostic classifications, and also identify disease subsets with distinctive pathogenesis, and then the identification of somatic mutations. That means that one is refers, this is not refers to the point mutation. Of course, we have to do NGS to see the point mutations. But somatic mutation itself, this one is refers to any like losses or gains or whatever deletion regions that can be detected in our cancer patients. Okay, so a little bit introduction about single nucleotide polymorphisms. Single nucleotide polymorphisms, SNP arrays, high density oligonucleotide based arrays that can identify both loss of heterozygosity, LOH, at individual nucleotides and copy number changes. SNP array has been developed to test for genetic aberrations that may predispose an individual to disease development. And there are two examples here of common SNP array. From Affymetrix or Thermal, we call it a gene chip human array, and also human cytosNP array by Illumina. And SNP array is a powerful technique to detect genetic aberrations such as LOH and also copy number alterations related to deletion or amplification. And it can characterize also LOH due to UPD, uniparental disomy, also called as an complementary LOH, and also known as acquired isodesomy or AID. And of course, this UPD or AID, they are usually not identifiable by conventional cytogenetic analysis, and FISH studies also cannot be detected by array CGH. So SNP array is one of the tremendous achievement that can detect this phenomenon. As a result, a SNP array has been increasingly applied to hematopoietic neoplasm to search for clinically significant genetic abnormalities. And a large numbers of CNVs and UPDs have been identified in a variety of hematopoietic neoplasm. And CNVs detected by SNP array in some diseases or neoplasms are of prognostic significance. So actually this technology is is only used about 200 and 250 nanogram of a DNA sample. So what is array CGH? Okay, so array CGH measures genomic gains and losses, or identifies differences in copy number of genes involved in pathological states. And this array CGH uses actually patient versus control competitive hybridization. So it's like gains and losses compared to the control. So it's a bit different principles when you compare to the SNP array, because you need the normal controls and you put together mix, and then you put it on the glass chips, the microarray chips. So it can identify microscopic chromosomal abnormalities, submicroscopic alterations, including microdeletions, the microduplications, and unbalanced subtelomeric rearrangements. Array CGH also can identify the chromosomal gain, the chromosomal origins, and also size of unbalanced segments and also markers. However, this technique cannot detect balance alterations such as balance translocations. And it has been implemented as a clinical diagnostic tool. It's considered the first-line genetic test for selected cases. And array CGH actually shows better signal to noise ratios than SNP arrays. Thus, many of the duplications missed by SNP arrays actually can be detected by array CGH. And nowadays, it's commonly used for the evaluation of individuals with birth defects, dysmorphic features, and also a mental retardation. So I would like to share my first experience of using a SNP array when I did my PhD about more than 10 years ago. So I can just see like clearly differences, especially in data analysis and also the chip itself, because at that time I just used the 10K, only the 10,000 and also 50,000 markers on the SNP or 50K SNPs information on the arrays. But now it's already more than 1 million SNPs or 1 million markers that has been designed on the chips. So this slide shows some of my PhD findings and also from the collaborators. And in the year of this publication, actually, we were the first group that utilized 5 technologies to study the status of CDKN2A or P16 gene in childhood acute lymphoblastic leukemia patients. We use DHPLC analysis for mutation screening, methylation specific PCR for methylation status, and also SNP array and also array CGH and FISH for deletion status. And at that time, I used the ChinChip human mapping 10K and also 50K array from the FE matrix, and also 244K whole genome array for from Agilent. That one is for array CGH. So our array CGH actually is from Agilent. And our study revealed that the principal mode of CDKN2A inactivation was a genomic deletion. And FISH analysis is very important to validate the findings from the SNP array and also array CGH. And this study was done by our PhD student, Noliana, who has discovered interesting findings about this copy number variation among non-syndromic cleft lip and or without cleft palate with hypodontia. And this, Noliana used a new version of SNP array known as a Cytoscan 750K from FE matrix as well, and to identify genetic aberrations in these patients. And this high density array contains about 750K markers for copy number and approximately about 200,000 genotypal SNPs, which provide high resolution copy number, accurate breakpoint estimation, and loss of heterozygosity detection. And this figure shows the image of 26 kilobase loss at chromosome 3p 14.2, the regions which is overlaps with FHIT gene. Okay, so this is using the specific software, the CHAS software from the FE matrix from the Thermo. And also we found also the 6 significant CNVs included 4 specific regions of gains, and at chromosome 12q, chromosome 15q, and also chromosome 1p, while another 2 specific regions with losses at chromosome 3p and also chromosome 4q. So actually we can see a lot of things using this microarray. So, and also you can check whether is there any significant genes that are located on that specific regions that has been affected. Okay, so this result supported that CNVs significantly furnish to the development of these non-syndromic cleft lip and without cleft palate with hypodontia. So this is also another collaborative research with a reconstructive unit, science units. This work was done by our PhD student, Nurul Shazana, who is now working as a lecturer as a reconstructive unit. And we use a DNA microarray from Illumina to do a genome-wide linkage analysis in non-syndromic cleft lip and palate of Malay population. And this disease occurs as a result of multifactorial determinants involving both genetics and also environmental factors. So several candidate genes actually has been reported to play a role in orofacial clefts, including MSX1, SATB2, FGF, and also IRF6. So we were interested to discover evidence of linkage at chromosome 2q, particularly SATB2 gene in non-syndromic cleft lip and palate of Malay population and also its methylation status. So actually we used for DNA microarray to do the genome-wide linkage analysis, we use Illumina Infinium Human Linkage 24-bit chips. Okay, so this is one of the bit chip from Illumina. And our findings show that linkage evidence and significant low copy number of SATB2 gene in this patient, and confirm that genetic factors play a major role in causing cleft defects. And we also noted that epigenetic testing found this gene also was unmethylated, so actually could not support the epigenetic occurrence in causing craniofacial deformities. And our genome wide linkage analysis also found copy number variations of 5 genes in 4 large extended families that showed linkage evidence during using qPCR as for a validation purpose. And also our novel findings revealed the low copy number of this TOTS3 and also CO is called 21A1, okay, which may represent as contributing genes, and increase the risk toward non-syndromic cleft lip and palate formation in the family trees. Okay, but because in other studies, it has been reported that COL2A1, COL11A1, and COL11A2 actually were associated and influenced the risk of non-syndromic forms of cleft palate. So it has been hypothesized that the reduced induction of TOTS3 triggering FGF dysregulation in progression of lip and palate fusion, and finally drive the abnormal growth. So this one also we use the Illumina Infinium Human Linkage 24-bit chip. So we also use a cytoscan, a 750K array to perform a chromosomal microarray analysis in these 2 chronic lymphocytic leukemia patients. And based on the manufacturer, cytoscan 750K was designed to provide the most comprehensive coverage and highest performance for detecting chromosomal aberrations in a broad range of sample types of constitutional, cancer, stem cell, and also neurodevelopmental applications. And actually CLL is a type of leukemia which is very rare in Asian but commonly found in Western population. So it is interesting to know if we can detect any unique genetic aberration in our Malay patients. And currently we managed to have these 2 reports of 2 patients, and both of them showed a similar findings of chromosomal deletions on chromosome 13q 14.2 until q 14.3. Okay, and also both of these figures represent part of the whole genome view for copy number variation analysis. And this female patient actually had a 4.7 megabase deletion on chromosome 13q14, while this male patient had 1.07 megabase deletion at the same region. And it has been reported that deletion of chromosome 13q comprises of 50% of common cytogenetic aberration in CLL. And actually it is a good prognostic factor if detected as sole abnormality. So both articles in this slide highlight the application of microarray in diagnostics, and microarrays are currently a key tool in genetic diagnosis, allowing clinicians to identify specific subtypes within an overall disease category based on differences in gene expressions. Okay. And moreover, clinicians can also use information from microarrays to determine which treatment methods which most likely yield beneficial results for particular cancer patients. And diagnostic DNA microarrays also would be beneficial, okay, for the applications in cancer patients, for example, and also in other disease. Okay, so this is also shows some of the DNA microarrays for diagnosis diagnosis of the disease. For example, like in the genetic disorders and also the infectious disease. Also, we can use the microarrays for the faster detection of the microorganisms. So what is a chromosomal microarray testing or CMA testing? So it's usually used in disease diagnostics. So CMA is a molecular cytogenetic method for analyzing copy number variations. And this testing actually offers the capacity to examine the whole genome on a single chip with a resolution which is at least tenfold greater than the best G-banded chromosome analysis or cytogenetic analysis. So this actually can be considered actually as a diagnostic test, and it has been recommended by these societies, for example, like from the American Academy of Neurology, American College of Medical Genetics, American Academy of Pediatrics, and also International Standard Cytogenomic Arrays Consortium. And CMA also is considered as a first-year test for patients with developmental delay, autism, and also congenital anomalies. So CMA actually includes both array CGH and also SNP arrays. So this is one the best examples of this chromosomal microarray test, or this is called a cytoscan DX assay, cytoscan diagnostic assay. Is a qualitative assay intended for the postnatal detection of chromosomal copy number variants in genomic DNA obtained from peripheral whole blood in patients referred to chromosomal testing based on clinical representation. So this is actually the first and the only FDA clearance or FDA approved chromosomal microarray test to aid in the identification of the underlying genetic cause of this developmental delay, intellectual disabilities, congenital anomalies, and also dysmorphic features in children. Okay, so actually it has been reported that it has been approved by FDA in 2014, where a year before that actually Heatmaps or actually file the application to be cleared by the FDA. So this cytoscan, okay, is a microarray. The SNP based on the SNP microarray is to analyze the entire genome with only one test. Okay, so it has about almost 2.7 million markers on this chip. And this is another example of CMA testing actually developed by Centogene company. And this microarray also enables detection of known and novel structural aberrations, such as copy number variations, chromosomal imbalances, regions exhibiting loss of essence of LOH, uniparental disomy, and also mosaicisms. Okay, so this is just an example of this array that has been developed by Centogene company. Okay. And this is also the examples of gene microarray base. This is based on the gene expression profile. So MamaPrint, which has been developed by the Agendia company in Netherlands. So MamaPrint is a prognostic and predictive diagnostic test for early stage breast cancer patients that assess the risk that a tumor will metastasize to the other parts of the body. So actually this MamaPrint test analyzes the 70 most important genes associated with breast cancer recurrence. So it's a microarray base, also FDA cleared, and molecular diagnostic assay that assigns tumors into categories, whether it's high or low risk, and also is combined expression of 70 genes. Okay, and the test report MamaPrint provides a clear whether it's low risk or high risk result to better predict whether breast cancer recurrence can happen or not. Okay, and this is the woman who benefits. So the criteria that are who are the breast cancer patients that can be undergone with this MamaPrint testing. So array CGH is also commonly used in both research and diagnostic testing. And actually, who is the candidate for array CGH? Of course, patients with or without prior genetic workup who have mental retardation of unknown etiology, congenital anomalies, dysmorphic features, and a suspected syndrome with normal chromosomes or subterranean fish or other studies or stillbirth or fetal demise. So that means this actually can be done using array CGH. And does array CGH detect point mutations? So of course, no. Okay, so CGH cannot detect point mutations. You have to use sequencer to detect point mutations. And then array CGH also, you have two different kinds of array CGH, whether it's a whole genome and also it's a targeted array CGH. So targeted array CGH, actually you can just customize your array CGH based on the chromosomes, based on the genes that you want to study. And this is the examples of the array CGH, SurePrint and also SurePrint G3. And this is just only the different, like we have the 244, that's the maximum for the SurePrint HD arrays. And it's up to 1 million markers actually on the arrays. Okay, so now we are going to the last part of the presentation. This is related with the microarray data analysis. As we all know that microarray data analysis is the final step in reading and processing data produced by a microarray chip. So of course, the role of bioinformatics in DNA microarrays is also very important. I think everybody knows about the bioinformatics contribution. So it's widely used at the different stages of microarray interpretation and designs, and also microarray data analysis actually using specific softwares. There are different kinds of specific softwares where the needs the license also, or maybe it's like a free, you can just download from the website. Okay, so analysis of microarray data is very important to make sure that the huge data can be analyzed using appropriate softwares. So as we all know that microarrays can be used in many types of experiments, including genotyping, epigenetics, okay, for epigenomics. Also you can use the microarray, and translation profiling, and also gene expression profiling. So we have the first step actually, the process of the converting the scan image of the microarray into quantifiable or computable values and annotating it with the gene's ID, sample names, and other useful information. So we call this process as a feature extraction, okay, feature extraction. So, and after the feature extraction process, the data can be analyzed, okay? And array manufacturers often provide software to open and analyze their raw data files, okay? And these programs also may not always be available because may become obsolete after a few years, or may not be flexible enough for your needs. So there are several free software tools that can suitable for the downstream processing of the microarray files, okay? And for example, like we have the Galaxy's platform, the Gene Pattern, the GeneSpring. GeneSpring you need license to use the GeneSpring, and also the statistic of software R, okay? And this slide shows the microarray platform for gene expression profiling. This is just example like how the flow chart for the methodology in the microarray experiments. If you are using the gene expression profile, or whether you want to use the genotyping array. So of course, you need the samples, and then you put it on microarray slides, and then of course you have to perform with the processing, the hybridization, and also washing, and also lastly you have to scan your your chip, okay? And then of course in the gene expression array, you have the image analysis and normalization, and then after that the last for the data analysis, you can have like the identification of differentially expressed genes. You can do the cluster analysis, the classifications, and also other analysis. For example, that you can use pathway analysis and also transcription factor analysis. Yeah. So this slide shows the overview of the microarray data pipeline. So this is the feature extraction that I did mention just now, where your image, when you scan your chip, and then you have to convert it into the quantifiable or computable data, normally saved as a cell file or text file. Okay, and then you can do the quality control, whether you have to check whether your samples actually pass the quality control or not. If not, because there is a certain value, a certain point that you have to consider whether your samples is pass the quality control or not. And then you can do the normalization, and then you can see the differential expression analysis, whether you see whether the genes actually upregulated or downregulated based on the clustering. And then you can do the clustering analysis, and then you can do the gene set enrichment based on the gene ontology. And then of course you can do the pathway or network analysis, and then actually you can submit data to a public repository for sharing, for data sharing. Sorry. Okay, so this figure shows an example of a heat map in which genes have been grouped based on their pattern of gene expressions. Okay, so a common method of visualizing gene expression data is to display it as a heat map. Okay, so this is the example. So you can see the colors. You can have like a red and blue. Normally we can see red and green. Okay, and then you can have like a very dark, the black one. Okay, so you can have like a different intensity in color. So in heat maps, the data is displayed in a grid where each row actually represents a genes and each column represents a sample. Okay, you can have like a different sample. So this is just only a schematic diagram, only 4 samples, but of course in your experiment it can be up to 100 samples. And then the heat map may also be combined with clustering methods, which you can group genes and also samples together based on the similarity of their gene expression pattern. Okay, and then the color and intensity of the boxes actually is used to represent changes, not absolute values of gene expression. So in these examples, red represents upregulated genes, and blue represents downregulated genes, and black represents actually unchanged expression. So that means there's no changes in terms of the expression. And of course, it is very useful for identifying genes that are commonly regulated or biological signatures associated with a particular condition, for example, like a disease or environmental condition. So this is a gene set enrichment analysis and pathway analysis. So it is very useful for finding out if the differentially expressed genes actually are associated with a certain biological process or molecular function. Okay, so this is a common approach to interpreting gene expression data. So we use the gene set enrichment analysis, so or GSE analysis. So based on the functional annotation of the differentially expressed genes. Okay, and then the popular tools for GSE and pathway analysis include David. I think everybody heard about David. It's a free online tool, and GSEA also free. Ingenuity is license required, also Reactum, also free. And this is the example of pathway from Wiki pathway. That means from your gene expression microarray data analysis, you can have, and later on you can do your you can have your data and then translate it into the pathway analysis. And network analysis is complementary to pathway analysis and can be used to show how key components of different pathways interact. And then also this can be useful for identifying regulatory events that influence multiple biological processes and also pathways. So come to the end of my presentation as a summary. So from the summary, it is good to think or to know about challenges and opportunities from these microarray technologies, especially if you use this as a whole genomic approach. And some of the questions that we may ask, and how can we store the huge data sets generated? How do we handle such large data sets? And how do we clinically manage a variance of uncertain significance? And the large number of genes being tested may lead to a number of unwanted findings. such as risk factor for other diseases or to unclassified variants. Okay, so the data may include interpreting interactions between individual genes, and then storing big data is a huge challenge for which we need to be prepared with investment in bigger computers with the greater memories. And also, of course, we also need more bioinformatician in molecular medicine for developing statistical methods, and also to give an overview about the softwares that are very important for that specific data analysis. And of course, to help researchers and clinicians to interpret the big data set. And because some of the findings may not directly giving an answer to solve the complexity of the diseases, and costing of testing, especially for diagnostic, and limited grant budget may also give financial issues. Okay, and however, this whole genomic approaches may give opportunities in the development of these in the scientific study, like improve complex disease understanding. If more researchers doing this microarray technology, so we can have like so many information that we can help us to understand about the diseases. And for example, like a whole genome-wide association study for detection of rare variants association may help to improve complex disease understanding. And this also, this technology also may help to improve, like to address pharmacogenomics research questions. For example, like whether this drug may work for this particular patient or not. And the most important thing is the ultimate goal of translating genomic data into clinically actionable results that patients can directly profit from this technology. So it's almost 1 hour. So I just want to thank you to all my postgraduate students and staff in Human Genome Center, and also my collaborators. That's all. Thank you.
Okay, thank you, Associate Prof. Dr. Sarina, for a very informative talk. So now we open for a Q&A session. You please unmute yourself if there is any question to Dr. Sarina. Okay. Any question? Okay, Dr. Sarina, I have one question. Is it okay to ask?
Okay.
Okay. I mean, I'm interested on your last slide where you mentioned about big data. Okay, big data. So basically, last week we have learned about the NGS, and this week we learned about microarray. And barely we know that both technologies, they are very important in terms of generating big data.
Yep.
Dalam bahasa Melayu, we call it as penghasilan data raya.
Data raya.
You also mentioned about the role of bioinformatician, bioinformatics data analysis. This, right? Yeah, that we can chip in terms of analysis the data.
Yeah.
Because from your experience, Dr. Sarina, can you share with us about the current situation in our school, whether we have an expert that actually can help in terms of bioinformatics data analysis, and how actually, if let's say we want to do the microarray, for example, so what sort of thing that we should do? Is it we have to outsource the service, or to get the consultation, or we need to establish some sort of collaboration?
Okay, so thank you, Dr. Stinora. So maybe I can just answer the first one first. This one is related with bioinformatics. Okay, so actually we are trying so much efforts actually, because we want to get somebody as a bioinformaticians in our school. Okay, because actually the first from Human Genome Center, from our side, actually before that I have already allocated one slot for our RLKA, that we want one RLKA candidates to go for to further study on bioinformatics, but unfortunately we didn't have that candidates. Okay, So, and then of course RLK already like stopped for a while, and then we don't have that opportunity to get our own bioinformaticians. And then later on, we have our collaborators. At the moment, we have one collaborator from University Malaysia Sabah, Dr. Wan Fahmi. Already like in one of our research ground also, we have already collaborated with him. And currently, we have appointed a fellow academic. This is based on the USM budget under school PPSP. So we have appointed one bioinformatician from Qatar University. Actually, he is Malaysian, okay, but now working as an assistant professor in Qatar University. So we just appointed him actually starting early of this month. So we just had one virtual meeting with him. So I'm just we are going to have our first webinar soon, maybe end of this month or next month. So we want to introduce him to our faculty, to our PPSV faculty, so that we can have like anybody from USM actually, if you want to collaborate with him, so it's welcome for everyone to collaborate with him, because he has a lot of experience in bioinformaticians. Because we cannot just rely, because as for me, I cannot say I'm not the bioinformatician, so of course I cannot I can just only give an introduction about bioinformatics, but in terms of the technical aspects in the data analysis, we need the bioinformaticians. We need bioinformaticians because, of course, if you want to do NGS or if you want to do microarray, of course, we can just outsource. Yes, we can outsource if you don't want to do it by yourself, but we can outsource outside from the company. And then, of course, company will provide you the raw data or some of the basic superficial data. And sometimes the company will charge you if you want to do like further analysis for the data analysis. So actually, the major part of the process, actually the data analysis itself, whether it's a microarray, whether it's NGS, we need integrated, or we need like supports and also collaborations, and we need to have this engagement with bioinformatics to help us to understand about the data that you generated from this NGS or microarray technology. And because it's the very big data, because if you use a very high density of the array, or with the high density of the NGS platform, of course the data will be more like very big big data compared to the targeted approach. Okay. So is it answer your
Yes,
your
it
first
is.
question, Dr. Stinora?
Yes, it is.
Okay.
Great
And
to
the
know that
second part is
actually
the
you have a
I
fellow.
can't remember the second part, sorry.
Second part, I think you already answered all of my questions.
Okay.
Yeah,
So
yeah, thank you.
so because actually Human Genome Center got our microarray actually more than 10 years ago. It's almost 12 years ago, okay? When I just came back from the UK from my PhD, and then a year after that, we got the microarray. Because at that time, we use some of our colleagues also use we use the microarray for research, not for diagnostic. Because of course, if you want to use for diagnostic, because of the charge, because our patients in Kelantan normally cannot afford to pay about 1,000 or 2,000 ringgit for the genetic testing using microarray. So of course, we started with the research, and then so it's almost 12 years. And now our microarray is not cost effective to repair because it's not function now. So we hope that in the future we can have we can replace our microarray with a new system or with a new model of microarray if we want to proceed with the microarray. But of course, you can just still outsource if you want to use the microarray, and later on the important step actually the data analysis. You have to understand your data. If not, because the data is very huge, and there are a lot of things you can do using your data, the data from the genomic approach.
All right, thank you.
Okay.
Okay, we have a question in the chat box. Can I read it for you?
Okay.
Okay, it's from R.I. Samia. All right. The question is, what is the difference between NGS and microarray in terms of clinical application? Are they complementary or different?
Okay, that's a very good questions. So of course, the main difference between NGS is microarray is the purpose of the function itself, because NGS you can detect the point mutations or like very small deletions or substitutions or any kind of point mutations using the NGS. Okay, but NGS also they have the targeted and also the whole genome or whether it's a whole exomes. Okay, and then microarray microarray is like as I mentioned to you, is like a whole genome approach as well, but you cannot detect the point mutation, but you can detect the genetic changes, the genetic aberrations, whether it's a gene expression, whether you want to see the changes in the expression level, or you want to see the changes in the DNA level, whether there is copy number variance, or is there any gains, or is there any losses. Okay, you can see the segmental deletion or whatever. So it's like you can just translate it from the conventional karyotyping into the molecular karyotyping. Okay, it's like a molecular, you can see like very clearly starting from the chromosome number 1 until chromosome number, chromosome sex. But in terms of the clinical applications, yes, actually nowadays, if you are looking at nowadays, actually both can complement to each other, because but it depends on the disease itself, because for example, like in certain disease, actually microarray is the first year. Okay, you have to do the microarray first, and then later on you have to do the NGS. Okay, but it depends on the clinicians itself, whether what are they trying to see, or what are they trying to rule out. So whether is it applicable for the NGS or is it applicable for microarray. Okay, because I did mention to you a few examples of the microarray base. Okay, and then because I didn't touch a lot, because my presentation is not focusing on the NGS, but so I couldn't really give the examples on the clinical applications of the NGS. Okay, next question.
Next question is, can you advise us what is the best workflow if we want to identify novel biomarkers?
Okay,
I think you mentioned already, right? Should we start with DNA microarray or NGS?
Yeah, I will for my opinions, I think I will go for microarray if you want to identify for novel biomarkers, and that it depends on your the structure of your project, the structure of your experiments, whether you want to use the cell lines or do you want to use the human samples, whether you want to treat your cell lines with any natural products or any drugs, because of course it is rely on that, because normally we use the gene expression profiling if you want to do for novel biomarkers, but the SNP array also can be done as well. Okay.
All right. And then the next question also from Shazana Hilda. Based on your experience, what is the limitation in DNA microarray application compared to the other genomic techniques?
Okay, so the limitations I think would say it depends, it's based on the markers that we applied onto the chip, onto the array. Okay, because if you have like more information or more markers on the slide, you will be getting like unnecessary data, which is not maybe not applicable or unwanted findings that I did mention in my previous slides. Because if you have more data, and then of course you have like so many noises as well in the data analysis. And what else? The limitations, of course I would say the cost. Okay, I would say the cost. It's quite expensive if you want to apply for if you want to do as a diagnostic methods. For research, as actually it's quite because sometimes it's quite difficult to get grants if you want to apply for like 300K or 400K, because the cost, if you are using the DNA microarray analysis, is quite expensive. Okay, if you want to use a quite number of samples, but normally we started with a few samples first with the DNA microarray applications, but it depends on the budget as well. And other limitations, for me, of the data analysis itself, because it's quite challenging for us if we want to analyze it by ourselves, because we need bioinformaticians. I could say that the last words, as I think bioinformatics is very important in doing this microarray and also NGS.
Okay. All right. So the next question is more to technical, I would say. It's regarding to the QC in DNA microarray. Is there any standard reference genes that we could use as a positive control genes, such as qPCR that we commonly use, GAPDH, beta-actin?
No. The QC that I did mention, actually, this not related with the reference genes. Actually, the QC is the technical, is the process in the data analysis before you want to do for further analysis. There is a certain technical QC to check whether your sample actually is okay or not in terms of the quality of the samples and then also the quality of the hybridization signals, whether the process actually work or not. So there is no standard reference genes to be used in the DNA microarray, because normally we use the standard reference, actually the genome reference in the data analysis. That is also from the manufacturer, from our database. Yep.
All right. So another question also from Shazana. I guess Shazana is the one who really want to post with the microarray analysis next time. So how do we differentiate the non-specific binding on the microarray data?
Okay. I'm not really sure about the non-specific bindings on the microarray data. Whether are you referring with the non-specific bindings when you are referring with the hybridization process, where the fluorescence cannot be combined to our samples, to our DNA samples. But later on, it depends on the type of the array that you use, whether it's a genotyping array, or is whether it's the DNA microarray, or is the cDNA, whether you're using the gene expression profile. So sometimes it's very difficult to differentiate whether your data actually okay or not, but we have a certain limits of ratio. Okay, because the parameters or the analysis that we use for different arrays will have a different criteria, whether that signals actually we can consider as a good signals, or whether it's a bad signals. So because sometimes we call that as a background signals, the noises. So sometimes that one we can just get rid of. So there's a certain limit that we can, depends on our analysis, whether you want to put the certain threshold to make sure that the noises you want to get rid of. Okay?
Next question is, have we clinically used microarray as diagnostics and treatment management in HUSM?
At the moment, no, because of the budget. Okay, because of the budget, because at the moment we ask our patients, if the patient came to the USM, and then if that for certain criteria of the clinical presentation, if it relates with the like autism, developmental delay, the one that I did mention with the cytoscan DX, and also the RCGH, normally we suggested the patients to go to government hospitals, to KKM hospitals, because RCGH actually can be done in Hospital Tunku Aziza in Kuala Lumpur, Hospital Kuala Lumpur, to do that microarray, because that one just only they pay only 100 ringgit, because it's like the charge actually is sponsored by the government. So we cannot do in hospital USM, but we ask the patient, we directly ask them to go through under government hospitals, because we are not, we are actually the teaching hospitals. So we also can help them to send the samples through our lab, to send the samples to hospital Tunku Azizah in Kuala Lumpur to do for array CGH. Okay. Any more questions?
Okay. Another question, Professor Rina.
Okay.
From Muhammad Rizwan. In diagnostic microarray, is it necessary or compulsory for us to validate the microarray result with FISH or conventional cytogenetics or other modalities?
Okay. So in terms of the validation, normally it depends on the what we call that the findings that we found. Normally, if we found the novel regions or the novel operations, normally it is good to validate the microarray result with FISH. Yeah, normally we are using FISH or qPCR. If you are doing the gene expression profile, so you can do a qPCR to check whether that genes is really upregulated or not. But for the like a mama print, I don't think that you have to do the validation. I think because mama print is already targeted one, because there are 70 genes there, so no need to do the validation. Because when I check, normally they didn't do validation for the mama print. But if for array CGH, normally they use FISH. Yeah, with like a microarray or array CGH, normally we need to validate the results using FISH. Yeah.
Okay. Any more questions?
Dr. Sarina,
Yes.
Zana here.
Okay.
Okay. Actually, the question I received is the common question that I received from the postgraduate students. So those that working with the microarrays analysis, they having difficulties with analyze the microarray data later on. And according to what Dr. Nurasikin mentioned, is there any, bioinformatics experts in Malaysia that we can refer to if we having if we want to consult with the data analysis?
Yeah, I did mention just now, we have our collaborators, Dr. Wan Ahmad Fahmi from UMS, University Malaysia Sabah. Okay. You can just I can help you to if you want to contact him. And then now we have already appointed a fellow academic Dr. Rozaimi from Qatar University. So Dr. Rozaimi actually currently is our fellow academic in School of Medical Sciences. So actually he is the expert. He is the bioinformaticians. Okay, so we are going to introduce him soon. We are going to have the webinars. Okay, so we can collaborate with him, and then he's happy to help us in PPSP, because we USM actually pay allowance to him about 3,000 ringgit per month. So it's like a 1 year punya appointment. So during this 1 year, actually anybody, if you want to contact him, actually is advisable. Okay. And in Malaysia, Dr. Fahmi from UMS. Actually, he is one of our candidates to be the lecturers in Human Genome Center, but because of the technical problem, and then after that, he got offer from UMS before USM offers him. Okay.
Thank you for answering.
Yeah, you can just contact me for more details. If I can give you the contact details for Dr. Fahmi and also Dr. Rozaimi.
Okay. One more question, Professor Rina.
Okay.
Okay. You know that we are in pathology, so we normally rely on our samples that is based on paraffin embedded, PPFE, paraffin embedded samples. paraffin emitted samples. So I wonder, basically, can we use microarray utilizing this kind of samples, the FFPE samples?
Yes, of course.
And is it I mean, based on your experience, is it require a special technique to do this?
Yes, because whether you want to use the you want to do the gene expression profile, if you want to do the gene expression array, of course you have to extract the RNA from the FFPE samples, and then you convert it into cDNA. So the challenge there is to get the good quality of RNA sample from the FFPE sample or from the FFPE samples. But I think there is a quite a good commercialized kit if you want to extract the RNA from FFPE sample. And of course, if you want to use the SNP array or other type of DNA microarray, it's okay if you extract the DNA from the FFPE samples, because if you are using the SNP array, it's just only about 200 nanogram of DNA is we need that volume for one chip, for one sample.
Okay. Thank you, Professor Rina. Any more questions?
Assalamualaikum, Anilnya. Can you hear me?
Yes.
Okay, Professor Rina, thank you very much for the very informative talk. I just I'm quite new in this genetic area. I have two questions lah. The first one is very basic questions. You did mention in the first few slides regarding the terminology. You did mention on the genome, genetic, and genomics, right? There is another term that I maybe already a long time ago, but I discovered that they use the term epigenetics.
Yeah.
How about this the definition, or how we can apply this in the research? And anybody in PPSP is doing research in this epigenetic study. All right.
Okay. Thank you, from Hanim. Okay, so because I think we need another lecture for the epigenetics. Okay, because epigenetics, the difference between the genetics, because genetics you can have like a genetic changes. Okay, but the epigenetics refers to that actually your DNA sequences are normal. You can have a DNA sequences are normal, but the epigenetic happen because of the expression. Expression is changed, because this refers to one of the common process in the epigenetics is the methylation, is a hypermethylation. Okay, so we do have the microarray specifically for epigenomic studies. If you want to see the epigenomic, we call this a methylome. Is the if you want to see the hypermethylation profile in the whole genome. So there is a specific array for that. So epigenetics, that means your DNA changes, there is no DNA changes, okay? Because like a point mutation, of course, there is changes. But in epigenetics, your DNA sequences are normal, but the abnormal is the only for the expression. Because in certain region, in our genes, the genes, we can say the sequences of the gene is normal, but the gene we say is abnormal because the expression pattern is abnormal, because it's silence the gene. The gene cannot be expressed. So if the genes cannot be expressed, so there is no protein produced. So that's we call is the epigenetics. So one of the common process is the hypermethylations. That's the study that I'm also doing some of the methylation studies, hypermethylation studies in cancer patients. Yeah. Is it answer your question, Prof. Hanim?
Yes.
Prof. Farid also is one. Also is doing a lot of experiments in methylation. Yeah, epigenetics. Yeah.
That means for the methylation, you have to do this genetic study, right?
Yes.
Yeah.
We can detect the hypermethylation status using methylation specific PCR, or you can do viral sequencing, or you can do the microarray also. We can do for methylation. Yeah.
So among those three, which is the best method to do this hypermethylation study?
It depends on your samples and also the purpose. What do you want to see? Do you want to see the whole genome? Also, it's a targeted. If you just want to check only a few genes, you can just do MS-PCR, methylation specific PCR. And normally because the sample, we have to do the bisulfate treatment first, and then you can just go directly to DNA sequencing also can.
Okay, of course, in terms of the price compared to qPCR, this methylation PCR is more expensive, right? Or what?
Yes, in terms of the cost, if you just want to do only one gene, you can do MS-PCR, methylation specific PCR, because we just order only a few primers pairs for PCR. Yeah.
Okay.
Yeah. But for the validation, normally we have to do sequencing as well to confirm whether the methylation is a real methylation.
Okay. Okay, how about protein microarray? Is it available in genome
No.
USM in other schools?
I'm not sure in the USM, but in Human Genome Center, none of us are doing the protein microarray at the moment. No, yeah. And also, I don't have any experience using the protein microarray as well.
Okay, thank you.
Okay.
Okay. Yes. Any more questions? I keep on asking the same thing. So
just contact me if anyone to ask further questions.
Okay,
Later on.
yeah. So yeah, I think it's a wrap up, right, Dr. Sharina?
Yes.
So thank you,
almost well.
Dr. Sharina, for the insightful talk and updates in the microarray technology, which is have a main purpose in biomedical diagnostics. So I believe everyone here have gained more knowledge on the latest update in gene microarray and some key highlights in research and diagnostic. So ladies and gentlemen, we are coming to the end of today's session. On behalf of the committee members, we thank everyone for being here today and taking your precious time to join the webinar talk. So we wish you all a blessed day. So don't forget to join next week on the webinar discussing on enhancing online teaching and learning skill, which is we will post in the email later. So thank you very much. Oh, please don't forget again to fill in the attendance and also the feedback form. All right. And yep.
Yep. Thank you, everyone. Yeah. Thanks.
And also, yeah, we do have a recording session. So feel free to visit our Facebook page, Pathology, www.facebook.com/pathology.usm. All right. So with that, I end the discussion. I end the session for today. Thank you.
Okay. Bye.